In this video, we will discuss about this tumor-associated glycans. So previously or in our previous videos, we have discussed what are the glycans in general that are overexpressed on the cancer cells. And from our previous discussion, we know that one of the common glycans that are overexpressed on the cancer cells are the sialylated glycans. However, within those sialylated glycans, what are the different categories or what are the different bonds uh, that leads to the different categories of the glycans uh, that are overexpressed in the different kind of cancerous cells. So we will cover in general that what are the different kind of glycans. Not in general. In general means that we will not cover by cancer specific types, but we will cover by the glycan specific types in this video. So we will start with the sialylated glycans. So uh, two things or two structures that are overly expressed on the sialylated glycans are this structure, that is SLEX or SLEA, Lewis X or Lewis A, which are commonly known structures. Commonly known as this Lewis A or Lewis X structures are overexpressed on the tumor cells. So what are the difference between this Lewis A and Lewis X? It's the bond by which this fucose bind to this N-acetyl-galactosamine. Uh, in one of them it bounds to alpha 1,3 linkage, and another of them it bounds to alpha 1,4 linkage. I'm not able to correctly recall which one of them is alpha 1,3 and which one of them is alpha 1,4. So other kind of things that are observed, other kind of glycans, disrupt glycans that are observed is this truncated O glycans, and we have discussed about truncated O glycans. What are the commonly overall expressed truncated O glycans are this STN antigen, TE antigen, and this TN antigen. And differences are shown in this figure. The STN antigen, that is sialyl, TN antigen. Sialyl is attached to this GalNAc or N-acetyl-galactosamine, uh, or and this purple one is N-acetyl uh, galactose N-acetyl you can say Glycnac. This is Glycnac. I'm not able to recall the complete full name, and this yellow one is GalNAc, N-acetyl-galactosamine, N-acetyl galactose, or we can say that this is Glycnac. Let's consider it as Glycnac, and this one as GalNAc, yellow one. So these sialyl TN antigens are overexpressed, that has this sialic acid and binds to this GalNAc. And if there is no sialic acids, only this galactose bind, that it is known as T antigen, and only GalNAc is known as TN antigen. So these are overexpressed on the cancerous cells. And the other things are this because of the disruptions in the signaling pathways or change in the reprogramming within the cancer cells, it will also lead to this branching of this beta 1,6 glycnac that can be seen here, because of the over expression of this enzyme that is responsible for glycnac attachment to this polypeptide chain, and also because of the overexpression of the enzymes that are responsible for the fucose, it will leads to the bond formation of the fucose at this asparagine linkage that can be seen here in the N-linked glycans. The other things that are observed is one of the things that are observed is also observed is this polysialic acid. In this case, the alpha 2,8 linked sialic acids bonds are formed, and this kind of polysialic acid structures has been found in the cancerous cells. Finally, this we can see that this sometimes this glycophospho this uh, glycosphingolipids are also found in the cell surface of the cancerous cells, and it has different kind of glycan structures that have been characterized or nomenclatured differently. For example, these kind of structures are named as GM3, and simultaneously other structures are named as this GMA1, GDA1, GD2, GDB2, and GT1B. So I hope you got a basic idea of what are these tumor-associated glycans in terms of the different kind of sialic structures that are overexpressed on the cancerous cells. Or and if you need to understand more about these tumor-associated glycans and the role of glycans in the cancer progressions and migrations in detail, you can go through the reference that has been provided in the description. It's basically a review of the role of glycans in the cancers. That's all for this video. Thank you for your time.